Speaking of muscles and downstream, you're also involved in neuroprosthetic development in your research. Could you please elaborate on that a little bit more?
So, I mean, neuroprosthetics is both. It could be the thing where we record basically from the cerebral cortex, from nerve cells in the brain. Brain-machine interfaces are used to read out the patterns of activity from those cerebral cortex cells. And subjects, be it monkey subjects in the lab or being these days a few human patients in the US who are doing it already as humans, learn to control their environment based on the activity that they're producing with their cerebral cortex. Brain-machine interfaces. And we're doing this ourselves also. We have an animal who is used to work in a virtual reality, 3D virtual reality environment, and brain controls basically a cursor to move around 3D space. Mm-hmm. And the animal has learned a task similar to what I mentioned before, the center outreach task. It starts in the center and then moves the cursor to different positions, the corners of a cube. Now, this apparently is not a prosthetic device, right? This is just a cursor on the screen. Why are we doing this? We're doing it because one can use actually the logic of neuroprosthetics, this taking a set of neurons and their activity are now exclusively responsible for whatever action the animal produces. There is no other neuron that could have an impact on the action itself. Mm-hmm. And we use this as a tool for studying adaptation in neural circuits. We're using it as a highly controlled setting in which we can learn something about how the brain learns to control movements, where we have much more control over both who are the responsible neurons who are producing the movement and what is the type of feedback that the animal is getting. Because if the animal is not actually making physical movements, it's not changing anything of its somatosensory information. So the status of the muscles and all these signals that normally would come back when I change my body configuration, when I make a movement, this is basically held constant. And all feedback that the animal gets about the movement it produced is the visual feedback from controlling the cursor. Mm-hmm. And this we can control with a computer. Proprioception, the signals that come back from the muscles, we cannot control in an experiment very well. Mm-hmm. We can silence it maybe with drugs, but we cannot systematically shift it. Now, with this setting, we have a way of controlling exactly the feedback that the animals get and perturbing it in an experimental fashion. And we know who are the neurons who are producing this movement. And by this, we can basically apply perturbations and see how the motor networks change their dynamics to compensate for this perturbation. And that's the core mechanism in adapting our movements to changing environmental conditions and to changing sensory input. So we use it on the one hand at the cortical level as a tool for answering basic research questions. Mm-hmm. But as you mentioned, we're also working with clinical partners and partners from industry towards real prosthetic devices. I have to say in the recent years, we've paused those activities. These are activities that date back a few years. But back then our logic was, okay, highly invasive chips or microelectrode arrays, I prefer to call them, implanted below the skull in the brain is something where it's not really fully clear yet how well this will be accepted by patients, how eager they're to get those. If you listen to the people who received those implants in the US, they are super positively excited. At this point, I cannot say how representative this sample is, but in Europe, it's clearly from a political perspective, the medical field is much more reserved about this possibility of opening the skull, putting electrodes in, and using this as a control device towards controlling advanced prostheses. But what is very common already since many years is controlling limb prosthesis with muscle activity that is still available. So that, of course, would not work for a quadriplegic person, a person who is paralyzed in all four limbs. But let's say if you have an amputee who is missing the lower arm from some trauma, from some accident, and the rest of the body is still under the control of the patient, it still can send motor commands to other muscles, then you can pick up those motor commands from the muscles. And it's relatively comparably easy, at least, to record muscle electric activity. It's not as invasive as brain electric activity. You can record it from the surface of the skin. And that is something that is established since many, many years. It's in the clinic, you have a lot of patients who carry and wear those types of prostheses that are controlled by muscle electricity. The challenge that we back then were trying to address in communication with the companies who produce those products, they said that many of the patients are not so happy with it because the electrical resistance, the impedance between skin and electrode changes during the day when you're sweating, when the temperature changes. It's not the best signal to start with, and they were often frustrated about the impaired control that they have on the device. And you can imagine, if you're interacting in a social setting and the prosthesis is not exactly doing what you want, it feels uncomfortable. I took one of their prostheses once and were gluing myself electrodes to my skin. And then we went to one of these political events. It was an open house in the government in Berlin. And then comes the minister of Lower Saxony and wants to shake hands. And I took this prosthesis and was shaking hand with the prosthetic device. And I was so scared that it would press too hard and that I have not enough control. Now, of course, a patient would have much more opportunity to control that than I had. But still, you want this thing to function. Otherwise, if you cannot rely on it, you put it to the side. And that's what many patients did. They put it to the side and decided and opted for a purely cosmetic prosthetic. Now, so that's why we set out to improve that. And the idea was, okay, it's not completely uninvasive anymore. But if we go below the skin, directly on the muscle, we're not suffering so much from changes in temperature, sweating, and things like that changes the impedance. So we get a much better signal-to-noise ratio. We can probably control much better because we can go on individual muscles, and the lower arm alone for controlling hand has many, many muscles, right? And what you record at the surface is a superposition of many of those signals. So the idea was, let's go on the surface of the muscle, and subepimysial, it's called, for the aficionados, and implant chronically electrodes there. But then, of course, we had the problem, now we need to get the signals out to the prosthesis. And having a wire going through the skin is A, aesthetically not so appealing, B, it's a hygienic risk that there might be an infectious pathway. So that's why we said, okay, let's go for wireless and transmit the signals through the skin. And that's what we then tested. That's where we came in. The partners from different universities and from the company were responsible for building the electronics for the wireless transmission. And we were basically training animals, rhesus monkeys, to get those implants and do their goal-directed reaches. That's what we are experts in, to train this to them. That was not a big deal. And then test in how far we get good enough signals from below the skin to the prosthetic device. Now, back then, it also was not really a prosthetic device, but we do some offline decoding of the signal. And it turned out it's just much more stable over time than whatever you can record from the surface of the skin. So in that sense, it was very promising. And then the project went into a phase where I said, okay, if we're now talking about longevity of electrodes, optimizing, the implantable devices, that doesn't really need a rhesus macaque model. We can test this in other animal models, which are not as valuable as a primate, and that's why we decided together that for a while now it will be tested in other animals before we might or might not go back to the primate model. I do think the next stage is probably directly go to humans. I felt so promising that one should directly go to human.
Neuroscience and beyond. Know more. Get inspired.